Welcome to the Business of Biotech Summer Executive Sessions Edition, where we get up close and personal with the people who are driving biopharma innovation and their roles as leaders of new and emerging biopharma companies. Joining me today is Jeff Wolf, founder of Heat Biologics, a company that's built a proprietary GP96 platform to activate immune responses against pathogenic or cancer antigens. Jeff is a multi-time founder and venture capitalist, having previously launched or co-launched Seed One Ventures, Generation One, Tyrex Pharma, Elucis Therapeutics, and Avogen after beginning his career with the VC firm Castle Group. Jeff, welcome to the show.
Thank you,
Matt. It's a
pleasure. Yeah, really appreciate it.
Pleasure to have you. Did I get all those companies right? Is there anything that I missed? Kind of the high level view of your pre-Heat Biologics.
Yeah, you missed quite a few, but I don't talk about those. So I think all the ones that I talk about, you got.
All right, good. Well, if you don't talk about them, neither do I. So today we're going to get into the weeds a little bit about how you architected and continue to grow your current company around the GP96 therapeutic platform. But before we get into that unique perspective on the foundations of your company, I want to learn a little bit more about you and share that with the audience. So looking at your background, it occurs to me that you can do a lot of things, go in a lot of different directions when you walk away from NYU with your JD and earn a Stanford MBA, which you've done. So high level, why biotech? Where does your interest in science kind of lie? What's the genesis there?
I've always been interested in cool things, and even growing up, I always like new technology. I like learning about science. I like reading about things that just these crazy things that people don't really focus on. And when I graduated from business school, I really wanted to do something that was different at the time, something where I could constantly learn. So I got a job at a very early stage venture capital firm, but they were different from others in that they were focused really on forming new biotech companies at the time based on cool technologies at universities. And without much of a science background, it just meant I had to work 2 or 3 times harder than anyone else to really understand the science. But
I find myself in a similar situation right now.
Right, yeah,
as the new editor at Bioprocess Online.
Right. Yeah. I hear you. And it's, but it's something I really enjoyed, and I really enjoyed, keeping up to date on all the really cool new developments going on in science and in biotech. So I like it, and the fact that I really love the science makes it that much easier to form new companies based upon cool science.
Yeah. So having that VC background from the outset, what sort of what facilitated or inspired the jump to biotech founder? And was that a jump? I mean, did you consider that a jump, or did it just feel like a natural progression?
Yeah, it was more of a transition than a jump, because the firm that I worked for was not a classic VC form. I mean, everything they did, they formed from scratch. And that's really where I cut my teeth into how to go about acquiring new technologies and forming new companies around these technologies. And, I left and I started forming these new companies under the rubric of Seed One Ventures, which was initially I formed it with a partner. And eventually it was my alter ego in forming these companies, including Heat. But it set up kind of an organized structure for me to do this in.
Gotcha. Okay. Cool. So let's talk about heat for a minute. What was the genesis of heat? Uh, how did you kind of get that ball rolling?
Yeah, so at the time, I was looking at many new cool technologies under development, and I came across uh, this this really renegade professor at the University of Miami. His name was Dr. Eckhart Podak, and he was chairman of immunology there. But just a really brilliant man, and really a visionary. Uh, he's a man that really didn't care too much about the conventional ways that things are done.
Mm.
It really had he went where the science led him. He was a genius, and he was unstoppable. And I met him, and he really struck me as an individual who believed in what he was doing and was doing things that were really revolutionary. And he told me about this really fascinating protein called GP96. Now, GP96, a lot of work was done on GP96 at Sloan Kettering. It's a protein, and it's in all of our cells. And the natural function of this protein in our cells is to fold the other proteins in our cells and the peptides and place them on the cell surface. It's what's known as a chaperone molecule, GP96. It's a heat shock protein. Now, what they found at Sloan Kettering is that when cells die via necrosis or unnatural cell death, it releases this GP96 bound to the protein it's folding at the time. And over time, as the immune system detected this complex of GP96 bound to a protein, the immune system recognized that there's something wrong. It shouldn't be seeing GP96 out there. So GP96 kind of co-evolved with the immune system to become what's known as a danger-associated molecular protein, DAMP, or an early warning system to detect necrotic cell death.
Mm-hmm.
Now, when T cells, so GP96 is taken up by dendritic cells and cross-presented to T cells. Now, when the immune system detects this GP96, it goes into high alert because it knows that there's a problem. So it unleashes a very robust pan-antigen T cell immune attack against everything that it sees bound to GP96. Now, and what they found at Sloan Kettering is that this is the primary pathway to activate T cells due to necrotic cell death. And Dr. Podak looked at this, and he studied this, and he said, this can really be a very robust way to activate T cells against cancer, against infectious disease, against a whole host of other things. If you could kind of harness this approach, harness these hundreds of millions of years of evolution and create cells that continually secrete GP96 bound to antigens of interest. And that's exactly what Dr. Podak did. So he was telling me about how he harnessed this approach. He created cells that are continually secreting GP96 to activate T cells against cancer and infectious disease. Mm-hmm. And when I first heard this, I really didn't believe that an approach like this is possible. I mean, how can you genetically modify cells and activate these cells to genetically modify cells to secrete GP96 and just unleash a very vast pan-antigen, very specific T cell immune response against the antigens of interest. But he showed me the data, and we went through the data, and the mouse data was really incredible. And I was like, wow, this is really super cool, and it could be revolutionary and transformative against a whole host of diseases.
Yeah.
So I showed this to immunologists who I know that were a lot smarter than me. And I said, well, what do you think of this? They go, well, it's interesting, but whatever you do, don't form a company around it. I said, well, if it's so good, why shouldn't I form a company around it? They said, because everyone knows that the immune system is never going to, you're never going to be able to activate the immune system to treat cancer. And that was in 2008. And now flash forward, immuno-oncology is the hottest thing. It's one of the hottest things out there.
Yeah.
And it's good that I did not listen to the advice of experts.
Sure. Well,
that's when I formed this company.
Absolutely, and that's where I was going with my question on that. You characterized Dr. Podak as a renegade professor, and here you are, a venture capitalist, perhaps looking for your next opportunity, interested in this science. Sounds risky though, especially during that time when immunotherapy wasn't necessarily considered an opportunity, or there wasn't a future there for treatment of cancer.
Mm-hmm.
How did you reconcile that? How did you I talk with so many leaders of pharma companies and emerging biopharma companies whose primary MO, especially in the fundraising early stages, is to de-risk everything. But it sounds like there was some inherent risk in the opportunity that you were seeking with this quote unquote renegade professor and his crazy ideas.
Right. I mean, so I think that there are times when you listen to the experts, and there are times that you don't. I think great revolutions in science have come about in a large part from the renegades, from the people who have different ideas. And most of these ideas were dismissed by the more conventional scientists, the scientific establishment, for many years until the data was overwhelming. And this happens in so many different areas in science. So at some point, you have to dismiss the experts and go with your gut. And in this case, my gut told me that, and my review of the data that Eckert generated and his lab generated, and it told me that he's really onto something, and he's onto something potentially revolutionary and different. It's going to take years to prove out, but if this works, it could be transformative. So I went with it.
Yeah. Yep. So I have to ask, why Heat? It's an interesting name. Why Heat Biologics?
Because we heat up the immune system. We supercharge the immune system with T cells.
All right.
To activate a robust T cell immune response.
Yeah.
But it's basically supercharging T cells.
Cool. So you just said a minute ago, you were excited about the opportunity, the possibility, the potential, if we could make it work. So what was the most challenging thing about making it work, or getting to a point where you had even more confidence that it could work?
I think the most challenging thing, and actually the biggest mistake that I made at Heat was our first couple of clinical trials where we dosed patients as a standalone, use this as a standalone as opposed to with a checkpoint inhibitor. Mm-hmm. So what we do is, and what we do very robustly, is we activate T cells, but the T cells still need to get into the cancer. They still need to destroy cancer cells. And as you know, cancer cells are protected by this kind of milieu in the cancer microenvironment. And checkpoint inhibitors break down this milieu that's protecting cancer from immune attack. And so we went in as a standalone. We were generating T cells, but we weren't getting to the cancer. Mm-hmm. You have to combine with a checkpoint. And our drug helps checkpoint. It's synergistic. We believe it's synergistic with checkpoints, and checkpoints are synergistic, we believe, with our drug. But you really need that 1-2 punch. So I think by combining with checkpoints, we're now seeing data that's really amazing, and data that we recently announced at ASCO that show that we can, using our drug, we've doubled the overall survival benefit of patients with late-stage patients with non-small cell lung cancer compared to trials that were with checkpoint inhibitors alone. So in trials with checkpoint inhibitors alone that these trials were approved on with BMS's checkpoint inhibitor, these patients saw a median overall survival of about a year, about 12 months. And we're seeing a median overall survival when we combine our drug with checkpoint inhibitors of about over 28 months.
Mm-hmm.
So we show that we activate T cells, we show the T cells we activate and invade the tumor site. And now we're showing that there's potential and substantial overall survival benefit by combining our drug with checkpoint inhibitors.
Okay. So I want to talk about the structure of your company and its organization. Shift gears a little bit here and still discuss the GP96 platform, but it's my understanding that most, I guess, I don't know if most is a fair characterization, but we'll say most biopharma companies form an organization, perhaps around a molecule or an idea, but then they sort of fit the science of the organization. It's my understanding that you intentionally structured your company around this platform, and I want to understand that a little bit better. So tell me about that. What's the relationship between the organizational or management philosophy of your company and its structure and the platform that you developed?
So we brought on people internally. So we're at Heat and its subsidiary Pelican. We're at about 35 team members. We've brought on team members that are immunologists, that are cancer biologists, are people who really understand the biology of cancer and the immune system, and who really understand the importance of this GP96 mechanism and how T cells can dramatically enhance the treatment of cancer. So we've built a team to specifically develop this drug. And now we acquired a new drug a couple of years ago that we just announced a phase 1 clinical trial on that has a complementary mechanism of action. So the team that we've built really understand immuno-oncology. They understand how T cells can assist in treatment of oncology, and how these drugs can really augment treatment.
The Business of Biotech is committed to helping the leaders of new and emerging biopharma companies navigate the financial, organizational, human resources, and regulatory waters you'll encounter on your way from discovery to the clinic and beyond. Cytiva, formerly GE Healthcare Life Sciences and the gracious supporter of this project, is also committed to that cause. Check out Cytiva's resources for emerging biotech at cytivalifesciences.com/emerging biotech. That's CYTIVA lifesciences.com/emerging biotech. So it sounds like the foundational key is people, bringing in people and letting them sort of direct. And I'm curious, this new drug that has a complementary mechanism of action, was that sort of an ancillary discovery that maybe wouldn't have been found had you not had the right people on board, or had the right people on board to want to go out and acquire that?
Well, it's something we knew about for a long time and have been tracking this very novel target. It's a co-stimulatory target that can really supercharge our efforts, as well as checkpoint inhibitors as well. It's a co-stimulator, but it's a very novel one. It's a first in class. Mm-hmm. But I think if we did not have this team in place that really understood the science behind it, understood how to conduct clinical trials behind it, understood how to manufacture these drugs, we would not really have been fully able to take advantage of this opportunity.
Yeah. Team like that doesn't just fall into place. You've got to go out and make that happen. Tell us about that. How did you go out and assemble this team of folks who are well suited to work on this specific platform?
It took a while. It took a while to put together the right group of people to who really understood this. And we've made mistakes in the past in terms of hiring, and we've corrected those mistakes. But I think that the team that we have now is really the result of years of working with this drug, working with this platform technology, and really modulating what we need based on what we're seeing in preclinical and clinical trials with this.
Yeah. Are there any other, I guess, structural or, I don't know, maybe fundraising considerations or just key parts of the puzzle that you pulled together that were directly related to your intention to build the organization around the platform?
Yeah. Well, we knew that we're going to need a lot of money to develop this platform. Because of the potential ubiquity of the platform in a variety of different areas, and the cost of running oncology clinical trials in particular.
Yeah.
So we took the company public in 2013, and we've raised over 200 million for Heat already to develop this approach. I think the public markets really gave us access to the type of capital that we would need to really develop it, and we've raised money when we've needed to, and when the opportunities were there.
Yep. So 2008 founding, did you say?
Yes.
Yep. So 2008 founding, 2013 public. What as the company's grown, you said there are 35 associates at this point. And as you focused on building that team with very specific people with very specific, interests, intellect, skill sets, how would you, from the leader's perspective, how would you say that has paid off for the organization in terms of processing efficiencies and cost controls and responsibilities around the finances? Because it occurs to me that certainly that there would be an impact there.
Yeah, I mean, I think we've gotten a lot better over time. I think we now we have a great finance team that does their best to keep cost under control. But it's been a challenge. We have so many activities going on. We have activities that we've spoken about publicly with respect to HS110. With respect to PTX35, with respect to HS130, but also, and these are the drugs that are in clinical trials now. We also announced a COVID-19 program in March that we're working as hard as we can to move our product into the clinic for that using the same platform. But we also have programs we haven't announced yet, and these eat up time and resources and energy, but it's really going to position us for the future with these new programs. So, but the challenge is keeping cost under control, and that's something that is really in our DNA here. So we do everything we can. I mean, especially on the manufacturing and clinical side, to control cost and run things as efficiently as possible. And our finance team does all that they can to make sure that costs are within the realm, because costs really drive everything for a small biotech company like ours.
Sure. The finance team does everything it can, meaning keeps the pressure on, right?
Absolutely.
Keeps applying the pressure.
Yeah, I mean, we have departmental meetings. We where they run through cost and ask very difficult questions about why things cost so much. If there's any any steps we can take to reduce those costs. Even in our manufacturing as well. Manufacturing eats up a big chunk of cash. And
I'd say that's probably the case, in most biopharmas. But is there a specific aspect of manufacturing these particular therapeutics that exacerbates that cost of manufacturing challenge?
Yeah, I mean, these are biologics. So in the case of HS110, it's we're manufacturing living cells, and we're injecting those cells into patients. And so it's a very unique manufacturing process. Not that many people are really skilled in it. And in the case of antibodies, our PTX35 antibody manufacturing can be very expensive as well. So we work with groups that understand our unique needs as a small company and can work with us to efficiently manufacture our drugs for the clinic.
Yeah. So is all that activity outsourced, or do you do any manufacturing in-house?
All of our manufacturing is outsourced. When we thought about building our own internal manufacturing capabilities, but that's a very expensive proposition, and we could do it a lot more efficiently by outsourcing it, and that's what we do.
Sure. Okay. You mentioned that you're working on a COVID program as well. Is that something you can briefly touch on publicly?
Oh, absolutely. I mean, we've talked a lot about our COVID program. Basically, what we're doing is we're applying these very same tools the same vaccine platform that we've used to dose over 300 patients in oncology, GP96 platform. Oncology, we've dosed in preclinical studies with our collaborators. It's been used against the Zika virus, malaria, HIV, and NIH and Department of Defense funded preclinical studies. So what we've done is we've modified this GP96 platform to create cells that are continually secreting antigens of interest bound to GP96 against COVID-19. So we're looking to bring this program into manufacturing shortly and into clinical trials as soon as possible. But it's a very unique approach. And I think one way it differs from all of the other approaches that are in clinical trials. Right now, there's about a dozen companies that are in clinical trials for COVID-19, but all of them focus on activation of the antibody humoral response. We're focused primarily on activation of a T cell response, which, as we're learning now, is very important in the treatment, in the prevention of COVID-19. Yeah. So it's a unique program. It's a program, again, that we could use as a standalone vaccine, or we could overlay it on top of any approved vaccine to add kind of a T cell layer of protection. So it's a program we're really excited about, and we're working as fast as possible to get something in the clinic as soon as possible.
How challenging has it been for you? This is a softball. This is a lob.
Okay.
How challenging has it been? I mean, I know it's been challenging. I just want a story about it. So here you are. You're focused on HS110. It's making progress, and then COVID hits, and you realize that perhaps that's an indication that you can tackle. It's a race. It's a race right now from a market perspective, from a public health and safety perspective. Just tell me about how you've managed that challenge as the leader at Heat, allocating resources to keep your promising oncology programs moving forward, and at the same time jump into the race for a COVID treatment.
Oh yeah, no, it's been a challenge. It's been a challenge. I mean, we have a lot of momentum on our oncology side, and now, in March, we decide to add a COVID-19 program. So, we're working with groups on the outside to really help us manage that, especially our academic collaborators who are helping with the development and testing of the cell line and preclinical studies. But it's, I mean, we've all been working double shifts here to keep both programs going.
Double shifts and probably double shifts in a very disparate workplace, right? You've got people. I'm assuming you're not all, gathering at HQ and bumping into one another in the lab.
Well, yeah, actually, we have the lab. The lab is fully functioning
Oh,
now.
good.
We keep both labs open. So we have our main lab is in Research Triangle Park, North Carolina.
Mm-hmm.
And then we have a second lab in San Antonio, Texas. And both labs now, through this pandemic, have been fully functioning. So we maintain social distancing, and we take other recommended precautions. But it was important to us, I mean, especially as we're developing a COVID-19 vaccine, to ensure that both labs are fully functioning.
Right. Good. Well, that's good news to hear. I'm glad it hasn't disrupted lab operations.
No,
Or
it really hasn't.
Minimally anyway, right?
Uh-huh. Uh-huh.
Yeah, that's excellent. Well, we are running short on time, so I had more questions for you, but we're just about up to our end time here. But I do want to give you an opportunity to just share a little bit about what's next for Heat, and maybe share some concluding thoughts, maybe some concluding wisdom for the leaders of emerging bio companies like Heat.
Great. Well, we have a really active pipeline with everything going on at Heat, and I think the pipeline is augmented by some of the things we haven't spoken about, and we're not at liberty to speak about until we announce them. But
we'll regroup at that point. I'll bring you back on.
Yeah, there you go, and maybe you give me a full hour next time as well.
Yeah, for sure.
But the only the only thing that I'd like to add is this company wouldn't be possible if I would have listened to the conventional wisdom espoused by very well-meaning immunologists and others at the time. I think you need to, if it's something you believe in, if it's something the data shows, go with it. Don't listen to the advice of experts, because often experts are following what they know, and really doing what they believe is best, but it might not be the right thing for the science. So go with your heart, go with the science, and really do what you feel is best as you're forming the biotech.
Yeah, I just to ponder that for a minute, this combination of heart and science and discernment in where you want to invest your time, your money, your passion, and other people's time, money, and passion. It's inspiring, but it's a hard thing to wrap your mind around, like what that recipe looks like when you're making a decision that's rooted in sound and promising opportunity versus emotion and desire. How did you know? I mean, what was like the real, I guess the factor that really sealed the deal for you that this was going to be? I mean, Heat's been your longest, from my perspective, as far as I know, it's been your longest running project. It's the one that you really, went all in on from a bio startup standpoint. So I guess I'm just curious to kind of crystallize that a little bit more. How did you know?
Yeah, I mean, I really fell in love with the science here.
Yeah.
The science I really like the science on the other companies that I formed, but this one I felt was really special and can really have the ability to impact so many different diseases. So, and it's the kind of science, the more you think about it, the more obsessed you become with it, and the amazing things that this GP96 can do. And if you could harness this power, you really have the ability to treat many things and to impact many different diseases and many different lives. So so I decided to really go all in on this. And yeah, it's really, it's really fascinating, and I learn more about the science and tweaks on it every day.
Yeah. Well, keep up the good work, Jeff. It's been a pleasure to have you. Thanks for joining us.
Well, Matt, thank you. Really appreciate it.
Yep, and we will do it again sometime. But in the meantime, that's Jeff Wolf. I'm Matt Pillar, and this is the Business of Biotech, produced by Bioprocess Online and graciously sponsored by Cytiva. You can access more resources for emerging biotech at cytivalifesciences.com/emerging biotech. In the meantime, subscribe to this podcast, give us 5 stars, and subscribe to our newsletter at bioprocessonline.com. Thanks for listening.